Imagine a factory filled with machines, tirelessly producing any product you can think of. You take your starting material, it gets processed and transformed into your desired product. Now, imagine a factory so small you can't even see it with the naked eye. Microbial cell factories are microorganisms that have been engineered to produce desired compounds by harnessing existing metabolic pathways. Bacteria, algae, and yeast have all been utilized to make molecules and proteins to produce pharmaceuticals, biofuels, therapeutics, and lots more. Cells are able to produce proteins and other molecules through the processes of transcription and translation. In these processes, genes are read, transcribed into mRNA, translated into an amino acid sequence, and folded into a fully functional protein. Cell factories can be used to produce native products, heterologous products, or a combination of the two. We have been harvesting native products from microorganisms for centuries. Baker's yeast has been harnessed for fermentation, and a secondary metabolite from penicillin chrysogenum is the widely known antibiotic penicillin. With advancements in gene editing technologies, cells have become programmable, with the design of cell factories becoming its own discipline. The programmability means we can insert genes of interest into cells to produce the desired valuable product. We can also use recombination technology to improve the cellular activities of the cell to achieve higher yields and more efficient processes in order to lower production costs and simplify product collection. To investigate how cell factories work, we'll be looking at two applications: biofuels and pharmaceuticals and natural product chemistry. Biofuels are fuels derived from plant, algal, or animal waste biomass. These fuels are more sustainable than fossil fuels, which are finite resources and contribute to global warming. Two alternatives to fossil fuels are biodiesel and bioethanol. The former is produced through the conversion of oils with the aid of a catalyst, and the latter is produced through the fermentation of crops high in sugar. The alternative is harnessing cell factories for biofuel production. These cell factories are easy to manipulate, can be scaled up, and do not contribute to climate change. Yeast has attracted a lot of attention for biofuel production as they can tolerate higher levels of alcohol. Yeast cell factories have been generated using xylose, glucose, and galactose as carbon sources. However, one of the major concerns of biofuel production is the requirement of crops with fermentable sugars. This creates competition as these crops are staples for human consumption. The alternative is using non-food related biomass as a feedstock, such as lignin, hemicellulose, and cellulose. However, wild-type Saccharomyces cerevisiae is not able to metabolize these biomasses. So what's the solution to this? Genetic engineering techniques have been used so yeast can utilize lignocellulosic substrates. By expressing genes for xylose reductase and xylitol dehydrogenase, yeast can produce ethanol at 6.8 grams per litre. While not as efficient as fermentable sugar feedstock, being able to utilize alternative feedstocks is promising, and studies are continuing to try and scale up these processes. Recombinant therapeutics is a fast-growing sector in the pharmaceutical industry and plays a vital role in disease management. Many pharmaceuticals are derived from plants, have low yields, have a difficult extraction process, or are synthesized by organisms that cannot be scaled up to an industrially relevant level. Recombinant therapeutics can result in quicker syntheses with higher yields and enhanced purity while being cost-effective and scalable. Insulin is one example of a therapeutic currently being produced by cell factories. It was the first drug to be sold on the market using recombinant DNA technology in 1982. Prior, the treatment of diabetes was with insulin produced in animal pancreases. Recombinant insulin is now made in E. coli, transgenic animal cells, and mammalian cells, but yeast is the preferred expression system. With the increasing incidence of diabetes worldwide, the requirement for insulin will heighten, hence the need for cell factories that can produce the drug. The insulin gene has had one amino acid change so that it can fold properly within the yeast cell. The gene is read by the cell's transcription and translation machinery, and the benefit of doing this in yeast is that it can undergo post-translational modifications before being secreted out of the cell, where it can be collected and used. In the same realm, cell factories are extremely important in relation to natural product chemistry. There are thousands of natural products that are used in medicine and industry. However, there are a number of issues concerning the supply of these molecules. The growing market means yields are not high enough to satisfy the current demands, leading to overexploitation and endangering plants in their natural habitat. Cell factories, however, can decrease the reliance on plants for these molecules and can potentially increase yield and access to sought-after molecules. Artemisinin is one such molecule that is being produced by cell factories. This drug is used in the treatment of malaria and is extracted from the plant Artemisia annua, also known as sweet wormwood. The demand for artemisinin and its derivatives is increasing globally due to the increasing incidence of drug-resistant malaria. The concentration of artemisinin in plants, however, is extremely low, resulting in cell factories being investigated as a potential solution. Artemisinin is now being semi-produced in yeast. The cell factory utilizes an engineered mevalonate pathway, a morphidiene synthase, and a novel cytochrome P450 monooxygenase to produce artemisinic acid, with these edits being integrated into the S. cerevisiae genome. The precursor can then be industrially converted into artemisinin, but even using cell factories for part of the synthesis can greatly decrease production costs and hopefully make the drug more accessible. Overall, cell factories will play an important role in our future by producing valuable products. With their potential for scalability, customization, and sustainability, they hold promise for addressing various industrial, therapeutic, and environmental challenges in the future.